Yeah, I guess we will have the videos from yesterday. We have them already. In fact, Tatiana sent them to me. So we'll make a decision about how we're going to make those available, either on OSF meetings or via our website. We need to have a little chat about that off air at some point. But we can make an announcement tomorrow so people who missed yesterday's sessions can catch up.
Lovely. Yeah, we've had a couple of questions about people wanting recordings.
Up to 150.
Okay, I think we can start.
Okay. Hello to everybody, wherever you are. I welcome you to the first talk session today. And this is a rank prize funds lecture. And our keynote speaker is Professor Pieter Roelfsema. Before we start, I just want to remind you that we have multiple channels of communication so that you can watch the talk via live stream on the Facebook. The talk is also being recorded, so you can replay it later. After the lecture, there will be an opportunity to ask a question or to make a comment. We prefer that you use raise hand option in Zoom as a way to ask question, or alternatively, you can post your question or comment on a Slack channel. And the last minute info is that we will continue to use Groudcon through the whole week, so you can meet with your friends and colleagues in coffee breaks. You can follow this link on Coffee in the online program. So this is the most important info. And now I invite Professor John Mullen to tell us more about the Lord Rank and the Rank Prize funds. Please.
A very curious path links Professor Roelfsema's lecture to a Yorkshire miller born in 1888, whose charity since 2007 has supported this lecture. Arthur Rank, J. Arthur Rank, inherited a substantial milling business, flour milling, from his father. But he was a devout man, and in the 1930s wanted to provide Methodist churches throughout the country with projectors for showing religious films. But he couldn't secure films of sufficient quality, and so he acquired a production company. Well, you've got a production company, you need a distribution company, and he acquired the Odeon chain of cinemas. And by 1947, this Yorkshire miller was dominated the British film industry. Think of all those films that begin with a gong, brief encounter, and one that's very redolent of Pieter Roelfsema's city, Operation Amsterdam. He also owned factories for making projectors and lenses. So when, in 1956, the film industry was in decline, it was natural for the Rank organization to make a deal with a small New York company that was seeking funding for a new kind of photocopying, and this became Rank Xerox. So reflecting Lord Rank's businesses, flour, films, and photocopies, the Rank Prize Fund's charity supports and rewards research in nutrition and optoelectronics, nutrition from the flour and optoelectronics from the Xeroxing and the films, and the eye and the visual system have been considered an honorary optoelectronic system. And Professor Roelfsema has taught us much about this optoelectronic system. He graduated MD in 1991. He's professor of at the Free University in Amsterdam. And since 2007, he's been director of the Netherlands Institute for Neuroscience. He magnificently combines physiological, perceptual, computational approaches to our field. His papers cover attention, working memory, plasticity, and the role of recurrent processes in the visual system. He very bravely tackles issues of central communication and consciousness that intimidate many of us. And I believe, too, he's actively working on visual prostheses for the blind. So it's my pleasure to invite Pieter Roelfsema to give the 2021 Rank Prize lecture.
So thanks a lot, John, for a very, very kind introduction. So let's see whether I can share my screen and have my pictures online. Can you see them? Okay, great. So this is the topic of today, perceptual organization and Gestalt perception. So I would like to first say that I'm very proud and grateful to receive this Rank lecture prize. And it's also very nice to talk about perceptual organization because I've been interested in it for a while. And this basically shows you what we are going to talk about today. So if you see a picture like this, you probably don't have a hard time to see that this nose and this leg is part of the same zebra. So this is perceptual organization at work. We need to group together image components that belong to the same perceptual object. And what I always wanted to understand how this is implemented in the brain, and that's what we're going to talk about. So this is an overview of what I'm going to talk about today. So first, I'm going to give a brief introduction on feedforward and feedback processing in the brain. And that will trigger some ideas, and we will test them first in figure ground perception. Then I'm going to talk about a process that I'll call incremental grouping. This work is largely done with synthetic images. And then in the last part of my talk, we will investigate whether this also happens when we view natural images. Let's start with the introduction about feedforward and feedback processing. So if we do perceptual organization, the visual system makes it hard for itself because vision really starts with fragmentation. Initial analysis in early visual pathways starts with small receptive fields, and neurons in those regions seem at first sight only to be concerned about what's going on at this small location in the visual scene covered by their receptive field. Okay, that's not how we perceive. We perceive wholes. So somehow there must be a powerful system that allows the visual system to glue back together all those image fragments that belong to a single perceptual object. And that's the thing that I've been really interested in. And what we tend to find, and I'm trying to convince you that this is the case, that this is implemented with what we call an incremental grouping process, where image elements of the same perceptual object are incrementally labeled with enhanced neural activity. Now, psychologists call this object-based attention. And I'm going to argue that you can only do this for a single image component at any one time. There can only be one incremental group. So if you want to know about another zebra, you have to shift your attention and repeat the incremental grouping process for another image component. Okay, so if this is the system, or if this is the image that comes into the visual system, it creates this labeled representation, and we would like to understand how this works. So in a lot of the work I'm going to present, we used macaque monkeys as a model system because studying macaque monkeys gives you a large degree of insight of what's going on in the human visual system. Now, this is from a famous paper already 30 years old by Velleman and Van Essen. You probably saw this. So it shows you all the cortical regions involved in visual perception in different colors. And from the same paper, here they also arranged all these areas into a hierarchical scheme. And we know that if the picture comes in, there are feedforward connections that propagate information from primary visual cortex V1 to higher visual areas. And the low-level areas extract simple features, areas at intermediate levels feature constellations of intermediate complexity, and at high levels feature constellations of high complexity that correspond to object categories. So there's this transformation from pixels into object categories. Now we also know that there are lateral connections between brain areas at the same hierarchical level, and feedback connections here in blue that allow for a recirculation of activity. And we believe that this happens a little bit later, and that during these recurrent processing operations, the incremental labeling occurs. Okay, and we would like to understand how this is implemented. Now, I will argue that in the brain, there are actually two types of processes that implement perceptual grouping. And the first I'll call base grouping. It's just another word for the tuning of neurons. We find neurons that are tuned to feature constellations, grouping together, say, a particular texture with a particular shape. Now, not all these possible feature constellations that you might want to group are implemented as base groupings, and therefore we need another system, a flexible system of incremental grouping, and that is implemented by this labeling process, which is also taking more time. So it's more flexibility, but at the cost of more time. Now let's look at base groupings. In recent years, we have started to understand quite a lot of how this is implemented in the visual system. And part of it is inspired by a revolution in convolutional neural networks that also translate pixels in images to object categories, right? You can't have missed this. And this is from a very nice paper by Yames and Di Carlo a couple of years ago, where they demonstrated or suggested that different processing levels of the primate visual system correspond quite closely to what's going on in these deep convolutional networks. And if you then look at the tuning in these convolutional networks at low levels, you find receptive fields that resemble the receptive fields of neurons in early visual areas. At intermediate levels, you find also feature constellations of intermediate complexity. And at high levels, you find neurons that are units, I should say, because we're talking about neural networks that are tuned to object category. Okay, so based on what we now know from artificial neural networks, we really start to understand what's going on in terms of base grouping. And what about incremental grouping? It turns out that there are also deep neural networks that are starting to become pretty good in image segmentation. So this is one of them. It's called UNet. There are probably by now better versions. UNet works as follows. It starts with an analysis of the pixels. Then there's sort of a feedforward pathways that detects the object categories. And then there's a feedback pathway that then highlights the pixels that belong to a particular object. Now, I must admit that these artificial neural networks for image segmentation are not as biologically plausible yet as they are for object categorization, but they're also not so bad. And if you take this image, for example, such a network might create what is known as semantic segmentation, basically highlighting all the pixels that belong to persons in a scene. It's quite remarkable that this can be done. And there's another variant of it that is known as instance segmentation. Now these different people in the scene are not only labeled as being a person, but there are also a separate label for each and every person in the scene. Okay, one label per instance. Now, I argue today that this is not happening in the brain of humans. We have only a single label, and that's the enhancement of neural firing rates. Okay, so that is one of the main outcomes of today's talk. Incremental grouping can happen for only one instance at any one time. That's what I'm going to try to convince you of. Now, let's go away for a while from natural images because they're very complex and not always well controlled. So let's make it simple and let's talk about Gestalt laws of perceptual organization. Now they come in two flavors. On the right side, you have the Gestalt laws that determine what is perceived as figure and what is perceived as ground. Now I took this slide from a number of previous studies. I probably don't have full references here. So these are our classics, Wertheimer, Kafka, and there's a very nice review by Johan Wagemans a couple of years ago. So as I said on the right side, those Gestalt cues that determine what is figure and what is ground. So small image regions tend to be perceived as figure. Same is true for image regions that are convex as opposed to concave, symmetric regions, and regions that are closed. Okay, on the left, we have Gestalt cues that determine what groups with what. So you have the Gestalt grouping cue of similarity. Items with similar features tend to group. So here, items with the same color. Items that are connected in each other's good continuation, that are nearby, and that tend to move in the same direction. Okay, and my promise is that by the end of my talk, you will have insight in how all these things are implemented in the brain. Okay, so this was my introduction. Let's now go to the meat of it, figure ground perception. So this is a display where you see oriented line elements, maybe even pixels, and it's kind of obvious that line elements that have the left orientation form a square that may even seem to be floating in front of a background with a different orientation. Okay, what is going on here? This is the Gestalt grouping view of similarity. So we group together image regions with the same orientation and size because the small image regions tend to be perceived as a figure. Now, my close colleague Victor Lamme, more than 25 years ago, demonstrated that neurons in primary visual cortex care about these displays. So he placed a texture in the receptive field of a group of neurons, primary visual cortex, shown in green here. And here is a figure on the receptive field, and here the receptive field is on a homogeneous background, but the image elements in the receptive field are perfectly identical. Okay, what he found is that there's an early response that is probably driven by feedforward input from the LGN, where neurons don't care whether it's a figure or a background, but a little bit later, then you get extra activity if those image elements in the receptive field belong to a figure. Okay, so we believe that this feedforward response is driven exclusively by information in the receptive field, which is identical. And it takes a bit of time, a bit of recurrent processing before a neuron in primary visual cortex starts to enhance its response if it belongs to a figure. Okay, this is the process that we really would like to understand. So I will be talking about how this works in the brain. Then I'll briefly review the role of attention. A big question in the lab has been whether this figure ground modulation in V1 matters for perception. And finally, I would like to investigate whether it depends on feedback connections from higher visual areas back to V1. Okay, so how does this work in the brain? So it's always good if you ask these questions to look at the classic psychologists. Now, here are a few studies that ask this question. And the consensus seems to be that there are two types of processes for figure ground segregation. One is a process that is sensitive to feature discontinuities that happen at the boundary between figure and backgrounds. It's a boundary detection process. And the second one is called region filling or region growing. It works as follows. If you select image elements with a particular orientation, say here the left orientation, then it might be a good idea to co-select adjacent image elements that have the same feature because they're likely to belong to the same perceptual object. Okay, but now we want to know how this is implemented in the brain. And if you look at boundary detection, studies typically propose that to get this to work, you need iso-orientation inhibition. Neurons tuned to the same orientation should inhibit each other. So if you're a neuron and your receptive field is in the background, and you're tuned to the right orientation, you will be activated by the feedforward input. But your neighbors tuned to the same orientation are going to provide you with strong inhibition. Okay, suppressing your response. The same will happen in the center of the figure where neurons tuned to the left orientation get a lot of lateral inhibition. And only at boundaries, inhibition comes from only one side. So you get at boundaries a relative relief from this inhibition, thereby you get boundary detection. Now, region growing, and this may come as a surprise, requires the opposite connection scheme. And you now want to put excited tree connections between neurons tuned to the same orientation. So how does this work? Well, suppose you're a neuron tuned to the left orientation. There has been a cue at this location. You think your features are relevant to perception. So then you might want to tell your neighbors also tuned to the left orientation that they should also enhance their response because they're likely to also belong to the relevant object. Okay. Now we have a bit of a problem here, of course, because we want to make a neural network that can both do boundary detection and region growing, but we have opposite connection schemes and these effects might cancel. So the brain has to come up with a trick to solve this. And the trick that it seems to have found is to carry out boundary detection early on during the feedforward processing phase, and to carry out this region growing process at a later phase during recurrent processing. Okay, so we made this work in a neural network. So what you see here is primary visual cortex, so a map of visual space. Here are neurons tuned to the left orientation, they respond here to the figure. Neurons tuned to the right orientation here respond to the background. In the feedforward pathway, we have this ISO orientation inhibition scheme. So indeed, we get extra activity at the boundaries. And if you superimpose the maps, you see that there's a nice outline of the boundary of the between figure and ground with no region filling going on yet. Okay, now the trick is to have this scheme at multiple spatial scales, that is in multiple areas. Okay, in higher areas, receptive fields become larger. And here in V4, we will have neurons that have about the same size as the size of the figure. Okay, they are tuned to the left orientation, and their neighbors have their receptive fields on the background, so they will not be activated if they're tuned to the left orientation. So these neurons have very little lateral inhibition. You might call it pop out. Okay. Now, if you present a larger figure, pop out is predicted to happen at a higher hierarchical level. And we're going to use this focus of enhanced activity in the feedback pathway to also fill in the center of the figure with enhanced neural activity. Okay, so this is region filling. Now, this is a model. What is nice about it is that we know from psychophysical work that boundary detection and pop out are known to be pre-attentive. And the prediction from this model is that the region filling might depend on attention because it depends on this loop through the higher visual areas and back. Okay, brings me to the next topic. What is the role of attention in figure ground segregation? Now, to find it out, we trained monkeys on a task in which they saw a figure ground display. I hope you can see the figure. They were supposed to make an eye movement to the center of the figure. This gray zone is just an example receptive field. And on some of the days, that's the only thing they had to do, make an eye movement to the center of the figure. On other days, they could completely ignore the figure. They only needed to care about the curves, and they had to mentally trace a curve starting at the fixation point to locate a larger red circle. Okay, this work has been done by Jasper Poort several years ago. And basically on figure detection days, the animals were making an eye movement to the center of the figure. On curve tracing days, they could ignore the figure. And if this is a receptive field of neurons in primary visual cortex, they could fall in the center of the figure. But of course, we could also displace the figure so it would fall on the edge or even completely on the background. Okay, so what we observed was basically a replication of the effect that Victor Lamme saw many years before. So image elements that belong to a figure here and red and green, blue and green, elicit more activity than image elements that belong to a background. Now, in this study, we were particularly interested in these excess spikes that are fired if the receptive field falls on the figure. Okay, so we basically subtracted the background responses from the figure responses to get this complicated 3D plot, where here on the x-axis you have the position of the figure which was larger than the receptive field relative to the receptive field. So at 0, the receptive field would be in the center of the figure. At +2 degrees, the receptive field would fall precisely on the edge. At 4, it would be on the background, and on the other side, the same story. Here it would be at -2 at the edge and at -4 at the background. Now these are these excess spikes, and you may notice that the excess spikes start at the boundaries. And then you have a later filling in of the center of the figure with enhanced activity. Okay, so early boundary detection, feedforward processing, and then later filling in of the center of the figure with enhanced activity. Now, this was in the condition that the animal was interested in the figure. So I would like to invite you to have a mental picture, what you predict what the situation would be if the animal would not be interested in the figure. So here's the result. So you basically still see the boundaries, but you see that this region filling in process is much less complete. Okay. Now we carried out also some recordings in area V4 where receptive fields have about the same size as the size of the figure. And there you see that in the center of the figure, the response enhancement is there early on board. And we also have some evidence that if we silence V4, you get a bit of a collapse of this figure, a region filling in primary visual cortex. Okay, let me summarize this for you. So you present the figure ground stimulus. V1 neurons have a map of visual space. They become active, start to code for the different features in their receptive fields. A little bit later, you get an enhancement of those receptive fields that happen to fall on boundaries. And it doesn't really depend on whether you're interested in the figure or not. And this is then followed by a phase where also the center of the figure is filled in with a response enhancement, but mainly so if you're interested in the figure and much less so if you're not, as if these non-attended figures are left in a somewhat more primordial state. Okay, so early boundary detection is pre-attentive. The later filling in of image regions depends on attention. Now, this was a big question for us. Is there a causal relationship between these response modulations we see in V1 and perception? And that turned out to be a hard nut to crack. We tried manipulation methods such as optogenetics in monkeys, but we found it very difficult to get it to work. So we now are going to switch species. So this is figure ground modulation in monkeys. Actually, we sometimes are in a very happy position to be able to record single units from humans in a collaboration with epileptologists. This was a patient whose epilepsy always started with visual hallucinations and who got an electrode so that we could record actually spiking activity in area B3. And also there we saw that figures here in red elicit more activity than the background. Okay, but for the purpose of causal manipulations, we are going to go to the mouse, where V1 is also present. And the happy news is that also in the mouse, you get figure ground modulation. So these animals can detect figures and have similar responses in primary visual cortex. So this was first discovered by Matthew Self, but then later really worked out also the perception of it by Ulf Schnabel in the lab. So these are the stimuli that we're presenting to the mice. And they have a very simple task, a localization task. If the figure happens on the left side of the screen, they make a lick response to a licked left spout. And if they see a figure on the right, they make a response licking to the right. Okay, so they can do this. And this shows you their accuracy. So here you don't need any figure ground segregation, just contrast defined figure on a uniform background. And the animals like that best. They're about 90% correct in the licking. And orientation in phase and texture defined figures. Ground stimuli, they're a little bit worse, but clearly above chance level. Okay, so we can also do the recordings in the mice. And basically what we find is very similar to what we find in the monkeys. So this is contrast in the receptive field in green or nothing in the receptive field in black. So this is just the visual response. You can measure the latency by subtracting the two responses. So visual response latencies in mice, here are 60 milliseconds or so. Now here are orientation defined figure ground stimuli, and you see that you get more activity elicited by figures than backgrounds at a latency of 95 milliseconds. And the latency for phase and texture defined figures is just a little bit longer. And now the really cool experiment. We're going to manipulate, we're going to completely silence primary visual cortex. And this is work that has been carried out excellently by Lisa Kirchberger. So we're going to use optogenetics. So if this is the visual response elicited by figure and grounds, we use optogenetics. There's an optogenetic silencer known as GT-ACR2 in which we can knock out all the spikes. And we can basically knock out the entire visual response in primary visual cortex. We can also, by shining the light a little bit later, let through the feedforward response and selectively block the recurrent processing phase, or let through more of that phase as well. Okay, so that's what we're going to do. Here is the contrast detection task, so no figure ground required. It's a bit of a complicated plot. Here you see the accuracy of the mice. So different gray symbols refer to different animals. The green is the average performance. So this is when we don't shine any light. So the animals are about 85% correct. Now, if we start shining the light at 30 milliseconds, we basically knock out all the visually driven activity. And you see that the mice, they have a decrease in their accuracy. It's about maybe 65%. It's not all the way back to chance level, which is 50% here. So it may actually indicate that there are some subcortical pathways, maybe through superior colliculus, that support some of this accuracy. Now then we start postponing the light, and you see that once we have postponed the light to about 70 or 80 milliseconds, the animals are back to ceiling performance. Okay, so apparently just letting through the feedforward response is sufficient for the contrast detection task. No need for recurrent processing. The situation was different for the orientation-defined figure ground task. You see that now if we silence V1 entirely, the animals are really at chance level, so they do need V1. And we have to postpone shining the light to about 120 milliseconds before they're back to ceiling performance. Okay, so they do need a little bit of this late processing phase during which we see the figure ground modulation. And we have to postpone the light by even more in the phase-defined texture segregation task. Okay, so this is excellent news. We see that if we only let through the feedforward response, the animals can do contrast detection. But for figure ground segregation, they really rely on this recurrent processing phase in primary visual cortex, causally linking this late processing phase in P1 to perception. Now, a small adjustment of this experiment also allowed us to ask the question whether the figure ground modulation in V1 depends on feedback connection. We now simply injected the optogenetic silencer in higher visual areas. Now, the mouse is a bit different from the primate in that the higher visual areas are arranged in the ring around primary visual cortex. And we knocked out quite a number of them. So this shows you the gene expression. And now we're going to go to the orientation-defined figure ground task, and we're going to do single unit recordings from primary visual cortex. Okay, and if you don't shine the light, we get the peak response and then the figure ground modulation as expected. But if we silence the higher areas, actually we're silencing them for a long time, you see that the peak response driven by the LGN is still there. But the figure ground modulation is much reduced. Okay, and that is also true if you look across a population of recording sites across a number of mice, that we knock out most of the figure ground modulation if we silence this ring of higher visual areas in the mouse. Okay, so we see that figure ground modulation, as we suspected, indeed depends on feedback from higher visual areas. Okay, so I've been talking mainly about the Gestalt grouping queue of size, because I showed small figures on a larger background. What about these other cues that determine whether something is perceived as figure or ground? Now, to investigate that, we use these texture strips where you see image regions with an alternating orientation of the texture and then separated by boundaries. Now, if you want, you can see this as a figure or this as a figure, right? They're completely ambiguous. But you can kind of start to bias perception by introducing convexity. So now most of you might agree with me that this is now more figural than this became more background-like. Okay, so it's very easy now to place these image regions on V1 receptive fields. Is what we did in a study that has been carried out recently by Anne van Ham and Danique Jurissen. And if you then record from primary visual cortex, you see a little bit of response modulation. It's not much. It's significant, and we think it's not so much because in this particular study, we did not require the animals to do anything with the stimulus. So they did not have to care about what is figure and ground. Now here we introduced also symmetry and closure. So now the figure perception here might be even stronger than was the case here. And then we also see that the figure ground modulation in V1 also becomes more pronounced. And actually in V4, we find that it's even more pronounced. Okay, so more generally, we see that figural image elements are labeled with enhanced neuronal activity. So much about the Gestalt laws that determine whether something is perceived as figure or ground. So I will now direct my attention to Gestalt laws that determine what groups with what. Okay, so now we'll be talking about incremental grouping. And here are topics I would like to discuss. I would like to discuss the mechanisms that support Gestalt grouping. I will argue that this is an interesting process that is transitive and can sometimes be very slow. I'll discuss the role of higher visual areas, and I'll argue and demonstrate that there can be only one incremental group at any one time. Let's start about Gestalt grouping. So if you see these 4 bars here, you might agree with me that this groups with this by good continuation. Okay, so if you select this bar, then we might want to co-select this bar. If the enhanced activity starts here, we would predict that the enhanced neural activity in visual cortex spreads to the other bar. Okay, and that's probably not the case. If we rotate the bars by 90 degrees, now activity would spread from here to here. Okay, that's a very simple prediction that we tested in a study a couple of years ago. So this would be the receptive fields of a group of V1 neurons. And we present the 4 bars. And then we require the animal to select one of the more central bars with a cue, cueing the animal to make an eye movement there. Okay, so now we expect that there's enhanced activity here that will spread towards the receptive field. We can, of course, also cue the other central bar, and now enhanced activity will spread here, but that will not be measurable in the V1 receptive field. And we can, of course, also, so this is shown, same as shown here. Now we can also rotate the 2 central bars. And then the prediction is that it really shouldn't matter anymore whether we cue this bar or that bar for the activity in the receptive field. Okay, so this shows the result. It's very straightforward, but the timing is maybe a bit complex. So at time 0 here, we present the 4 bars. So this is the first time something strikes the receptive field. And then after 500 milliseconds, we present the cue that groups with the receptive field bar. You get more activity. And if you present a cue that does not group with the receptive field bar, you get less activity. Okay, so enhanced activity spreads from this bar to the receptive field if it's cued. Now, this does not happen if you rotate the bar by 90 degrees. Then there is no influence of which of these bars we cue. Okay, let me summarize this for you. So if you select a central bar, then enhanced activity will spread to other bars that are grouped by good continuation. That does not happen if there's no such grouping. Now we played the same trick for color similarity. So here is a yellow dot in the receptive fields, and we cued the animal to make an eye movement to an adjacent dot with the same color by making it slightly larger. Okay, what we then found is that activity spreads from the cued dot to the receptive field. And this did not happen if the receptive field dot and the cued dot had different colors. Okay, grouping by color similarity. And the same for common fate. So it's a small manipulation, basically the same experiment. We cue one of the central bars, and we see that activity spreads from the central bar to the receptive field bar. And this does not happen if these bars are kind of moving in different paths. Okay, again, demonstrating that the enhanced activity spreads according to the Gestalt grouping cues. Okay, so enhanced neural activity spreads from cued items to items related to them by good continuation, similarity, and common fate. So this is basically going in the direction of explaining what's going on the left side of this slide. But now I would like to go into something that I find really exciting about perceptual grouping and perceptual organization, that it's transitive and slow. Okay, and I'll explain to you what that means. So if you see this display and you focus on the yellow dots, you may agree with me that it tends to group with the blue dot because they are close together. But now I'm going to add image elements, and you see now it's different. Right now the yellow dot actually tends to group with the red dot. Now the reason is that the yellow dot groups with this black dot, which in turn groups with this black dot, and so on and so forth until you reach the red dot. And this is basically another word, another explanation, or another way to express that grouping is transitive. If image element A groups with image element B and B with C, then A also groups with C. And as a result of this, to get the perceptual groups, you may have to evaluate quite a number of local grouping relationships, and this makes perceptual grouping also a slow process, at least in some circumstances. Okay, so here is the idea. If you are interested in the yellow bar here, this would spread to the yellow dot, it will spread to the blue dot, and here it will have to spread through a number of intervening links. Okay, now let's see this in reality. So this is a psychophysical task. This is more fun in front of a large audience, but we have to deal with Zoom today. So what I'm going to do is I'm going to present to you two curves, and one of these curves is going to make a connection between the red dot and either the green dot or the blue dot. So if you think it's the green dot is connected, you might want to raise your left hand, and if you think it's the blue dot, you might want to raise your right hand. Okay, and let's see what your reaction time is. Are you ready for this? 1, 2, 3. Very few people in the chat are raising their hand. I just can't see whether it's the left hand or the right hand. Okay, so you see this takes a bit of time. Let's start from the other side. So once more, we start now from the right. 1, 2, 3. Not bad. So typical response time in a large audience is 10 seconds or so. So it really takes you time to work it out, although all the information is there from time zero. So how do you solve this? Well, basically, you're probably going to direct your gaze here. And you're going to make a number of eye movements along this curve until you reach the blue dot. Okay. Now your eyes will typically remain on the target curve, the curve that you're tracing. So that means that before you make your first saccadic eye movement, you're actually going to do a bit of mental work, a bit of mental tracing. And then you're going to make your eye movement. A mental tracing, then an eye movement, mental tracing, and an eye movement. Now, of all the time you'll be spending on this stimulus, maybe 10 seconds or so, it's only 10% is eye movement time. 90% is mental time. Okay, so you've been spending 9 seconds looking at the stimulus to work out what groups with what. Okay, so we studied this several years ago in monkeys. So we trained them to look at the fixation point. Then they saw two curves. One made the connection between the fixation point and a larger red circle. This is the target curve. There was another curve as well, not connected to the fixation point. There was a distractor. And then after a delay, we removed the fixation point, and then the animal was supposed to make an eye movement to the end of the target curve. But we're going to record from the phase that the animal is maintaining fixation. He's only thinking about the curves, but not yet moving his eyes. Okay, this shows you an example of the recordings. This green rectangle was not visible. It's just a receptive field in primary visual cortex. So here the receptive field would be on the target curve. Here it would be on the distractor. This shows you the activity. So here, the first dotted line, the stimulus flashes on the screen. Something strikes the receptive field, you get a visually driven response, but that does not yet discriminate between target and distractor because what's going on in the receptive field is identical. You have to wait a little bit for the mental tracing process to get started. And then after a delay, you see that the target curve starts to elicit more activity than the distractor. So it's labeled with enhanced activity. And the same happens if the two curves cross each other. Okay, this is a recording from 5 groups of cells at the same time in primary visual cortex. Those that had a response enhancement are shown in yellow here. You see for all these configurations, the target curve is highlighted with enhanced activity. So here you see object-based attention at work. Now this is a very interesting case where we have 2 receptive fields. They're both on the target curve here. Here they're both on the distractor. Now, if you look at receptive field 1, you see that if you present the stimulus at time 0, first you get the visually driven response, always after 40 milliseconds or so. And then you get a relatively early enhancement of the response if the receptive field on the target curve after 106 milliseconds. Receptive field 2 is farther along the curve, so it probably takes the monkey a little bit more time before he reaches this receptive field. And you see also that although the visual response is of course the same, this response modulation is delayed after 375 milliseconds. Now, when we compiled data across quite a number of recordings, we came to the conclusion that we can best account for these results by what we call a Groudcon model of object-based attention, where the enhanced activity propagates over the curve, but not at a constant speed. The speed seems to depend on the distance between the target curve and the destructor. If they're far apart, it goes fast, as if the visual system is using large receptive fields so that you can cross a lot of space in just a single synapse. But if curves are nearby, you probably cannot use these large receptive fields because they would fall on both curves. Okay, so therefore, the system goes to lower visual areas with small receptive fields and now carries out the propagation in these lower areas. Now, in this Groudcon model, we assume that the size of the Groudcon is the size of the circle that is centered on the target curve and just does not touch the destructor. So we can then measure the distance between the fixation point and receptive field 1 as the required number of Groudcon shifts, which is about 1 Groudcon, and receptive field 2 would be about 3 Groudcons. Now here on the x-axis is the number of Groudcon shifts required to reach a receptive field. Here on the y-axis is the latency of the response modulation. And we get a reasonable regression with a slope of 50 milliseconds per Groudcon shift. So that means that if you have 2 receptive fields that are adjacent, just not overlapping, it takes you 50 milliseconds to go from here to here. Which explains, if you have more complex displays, like the very complicated curves I showed you before, these delays really start to add up, can add up to several seconds. Okay. Now, what about the role then of higher visual cortical areas? Because I was talking about different receptive field sizes. And to find out, we started to record from multiple areas at the same time. So here is V1, V4, and frontal eye fields. Frontal eye fields is an area of frontal cortex involved in the generation of eye movements, where receptive fields are larger than those in V4, which in turn are larger than those in V1. Here they would be all on the target curve. Here they would be all on the destructor. Now this is V1. We saw it before. The target curve elicits more activity in this delayed response phase. In V4, you get the same effects, maybe even a bit exaggerated. And the same in frontal eye fields. So you get first a feedforward processing phase that does not discriminate between target and destructor. And then later you get huge response modulation as the animal is going to select one of these items for an eye movement. Okay. So there's this late recurrent processing phase that you find in all areas, and interestingly also at about the same time. Okay. So the next time you are confronted in your kitchen with this complicated scene and you want to switch on the hairdryer, you now know there's a very simple algorithm implemented in your brain that allows you to pick up the right plug. Okay, and we believe this works as follows. So there's this mental tracing process that might start with large receptive fields in V4, but now there's this one receptive field in V4 that cannot do the job because it falls on multiple items. So V4 is blocked. Fortunately, there are feedback connections that make V1 and V2 also look smart and fast, because if V4 makes fast progress, these feedback connections will also seem in V1 that progress is made fast. Now, V1 is the lucky one with receptive fields small enough to carry out the process when V2 and V4 are blocked. At some point, of course, V2 can also kick in, and at some point, V4 can then also take over. Okay, so this is how we envision that this curve tracing process is implemented using multiple receptive field sizes. Now, we also looked at the perceptual psychology of it, and we basically find that object-based attention gradually spreads over such a curve. And this Groudcon model also gives a nice explanation of scale invariance. So this is actually from the great work of Pierre Jolicoeur and his collaborators who studied curve tracing at the end of the 80s and the beginning of the 90s of the previous century. This is one of their landmark studies. What they did here is present a curve tracing stimulus and then presented a version that was, say, 2 times or 3 times smaller. So the amount of curve that you need to trace is now 2 times or 3 times smaller. So you might predict a shorter reaction time. But of course, you can also only use receptive fields that are 2 or 3 times smaller. Okay, and it turns out that these effects perfectly compensate each other. So your reaction time on the left will be about the same as your reaction time on the right. Okay, that's of course well explained by this Groudcon model. Now, last point I would like to make talking about incremental grouping is this issue of how many incremental groups you can form at any one time. Now, to find it out, we asked subjects to detect curves that have 2 dots. Okay, and we showed pairs of curves or only one curve. Now, if you would be able to have multiple labels, you should be able to do that for multiple target curves at the same time. Okay, now, of course, we found that this is not possible. If you present 2 curves, you are much, much slower than if you present 1 curve, as if you can only do 1 curve at any one time. And to illustrate this a bit more, I made the following display. I call it DNA search. And your task is to find a curve with 2 dots on the same curve. Okay, and try to maybe raise your hand or in the chat if you see the curve with 2 dots. 1, 2, 3. Yeah, some people start to find it. Okay, it takes time, right? And I suspect that you find yourself scrutinizing each pair of curves or even a single curve at any one time. And at some point, you will stumble on the curve that has 2 dots. Okay, now this is, of course, very reminiscent of the work of Anne Treisman and her collaborators, who suggested that you need attention to bind features. But she was typically thinking of features like color and orientation at a single location. Here we're generalizing the idea because we're now not binding features that are different. We are actually binding features that are the same, but that are different spatial locations, right? So we're binding by spreading object-based attention. Okay, so there can only be a single incremental group at any one time. Now, why is that? Well, if you would try to use the enhanced firing rate for 2 curves, then you would erroneously bind those features as a single object. Now, it's also important for how we think about the neural code for perceptual grouping, because there's a competing theory that suggests that this is done by synchrony, neuronal synchrony, where image elements of the same object would fire, neurons that code them are firing their action potential synchronously, and image elements that belong to a different object would then be coded by a different pattern of synchrony. That was the idea. Actually, that was the idea that brought me to the perceptual grouping field. And I was very disappointed to find out that this is not the case. And I think by our finding that you can do this for only a single object at any one time also suggests that there is only a single label, so that's already evidence against synchrony. But we also did some recordings on the curve tracing task. And for those of you who are not so into synchrony, you typically measure it in a cross-correlation function, and the peak in this cross-correlation function is a measure for the degree of synchrony. So you have 2 receptive fields here that fall on the same curve, and you see that they're indeed firing synchronously. But you see that here are 2 receptive fields that fall on different curves, and the degree of synchrony is actually identical. And that's what we found when we also evaluated quite a number of pairs of recording sites. Okay, so really, synchrony is really not doing the binding. And our data are quite compatible with this propagation of enhanced neural firing rates. Psychologists call it object-based attention. Okay, so now in the last, let me see, maybe 10 minutes or so, I would like to see whether these results that we obtained with synthetic images, whether they also generalize to natural images. Okay, so we're going to examine whether we see incremental grouping when we process natural images. Then we're going to look in the brain whether there's also evidence for boundary detection and region filling. Okay, let's start with an incremental grouping. So we presented subjects with a task that is almost like curve tracing, and they basically had to look at images that always had pairs of vehicles or pairs of animals. And then we presented 2 cues, and the task of the subject would be to tell us whether they are on the same object, same truck, or on different trucks. And we varied the distance between the cues. So here they have distance 1, here they have twice the distance. And we also had a condition with twice the distance where the 2 cues fell on different parts of the same object. And of course, we had matching distances with cues falling on different objects. This shows you the result for the trucks think averaged across 15 subjects or so. And you see that reaction times are shortest if they're close together, a bit longer when they're farther apart, twice actually as far apart, and especially long when the two cues are in different parts of the same object. And this is also true when we average data across all the pictures that we had. Okay, so this is our first evidence that this incremental grouping process also happens when we're looking at natural images. And this is how we think that this is implemented by the spread of enhanced neural activity. Okay, now to further examine this, Danique Jurissen in the lab started to examine a bit more how we best explain how fast this incremental grouping process goes for natural images. So she started with these natural images. You see always pairs of animals or pairs of vehicles, but she also simplified it further by using cartoons. And here in these cartoons, you can probably recognize that there are lemurs or some kind of monkey here. So she also did a version that she called scrambled cartoons, where she displaced a couple of image components so that they're much more difficult to recognize. Okay, and we tested quite a number of models. I'm only going to review two of the models. One I call the pixel by pixel model, which suggests that this grouping process goes with a constant speed within an object. So you can think of a constant speed as receptive fields that all have the same size. Then, of course, we have our Groudcon model, where the Groudcon is determined to be so large. So if you start here, for example, that it is as large as possible, but is not allowed to go across object boundary. And then, of course, you get large Groudcons for these homogeneous large image regions, as is the case here in the center of the animal, and you get small Groudcons for the smaller parts of the animal. So you can work this out mathematically or in a computer simulation, and you can then measure the distance between a cue and any other location on the same object as measured by the number of required Groudcon shifts. You see that the distance between this cue and the end of the tail is largest in terms of the number of Groudcons. This is the task. And your subject is fixating, then he sees the two cues. Actually, one of them is always coinciding with the fixation point. And then the subject tells you whether these are in the same object or not. So a real image parsing task. And this shows you the quality of the prediction by the Groudcon model. And you see that it's quite excellent. So correlation coefficient of 76%. And so it seems that the Groudcon model is really doing a decent job here. And if you then look how much of the variance is explained, so the scrambled cartoons, the Groudcon model explains 58% of the variance, pixel by pixel model only 3%. So having the Groudcon really adds something. If you then go to the cartoon outlines that are recognizable, it not a lot changes. But you see, if you use the real life pictures, then actually the prediction is not so good anymore. So we're apparently not explaining all the variance that is to be explained for these types of pictures. And I think that's a challenge for us and maybe for others in the future. I think what's going on here, there are all kinds of interesting texture boundaries that may interfere with the spread of object-based attention, but there may be also some extra cues, maybe related to color similarity, that might speed up this incremental grouping process. Now, this is something to be explored in the future. Okay. So to summarize this part, indeed, we find that incremental grouping does occur in natural images. And one speculation that I have is that maybe that's a reason that we can only make 3 eye movements per second. So that means that we wait with the next eye movement for about 300 milliseconds. We know that feedforward processing is over after about 150 milliseconds. So that gives us another 150 milliseconds for incremental grouping and maybe to determine the next position of our eyes. Okay, so this was purely psychophysics. Now it's interesting to see how neurons in the brain respond to natural images with this idea of incremental grouping in mind. So let's first have a look at boundary detection. And here we took advantage of this Berkeley segmentation data set that many of you may know. And this is work that has been done together with the lab of Sergio Dumoulin and spearheaded by Paola Papale in collaboration with Wietske Zuiderbaan. Okay, so if this is one of the pictures these people in Berkeley, in the group of Malik, they asked subjects to annotate images and to highlight those boundaries that are particularly important for understanding the image, particularly those boundaries between central objects and the backgrounds. Okay, and here you see then one of these pictures that has been annotated. Now we asked a very simple question. Is there a difference in the response elicited by an object contour compared to a contour of equal contrast that does not belong, that is not so significant as an object contour? Okay, is this visible in primary visual cortex? Now we also did an fMRI version of the experiment. I'm not going to go into those results. So here I'm going to focus on the electrophysiology in monkeys. So you can require a monkey to direct fixation to a fixation point. And if this is the receptive field, you get a sample from the image. Then of course you can displace the receptive field, get a sample from another part of the image, and so on and so forth. So by having many fixation points, you can get basically the V1 picture for a particular receptive field of such an image. And that's what we did here. This shows you averaged across quite a number of recording sites, the average response in primary visual cortex by this picture. And you see that after 50 milliseconds, you get the visual response. Then the thing starts to elicit all kinds of activity patterns. And interestingly, after 300 milliseconds, you see that there's quite a lot of activity for the relevant objects and less for the backgrounds. Now, this is very poorly controlled because there are all kinds of also low-level differences between the objects and the background. Now, one of the most obvious low-level differences is contrast. Okay, so in this picture, we evaluated the root mean square contrast for all those image patches and sorted all the fixations based on the number based on the amount of contrast in the receptive field. So here there's a lot of contrast in the receptive field. Here's a little contrast in the receptive field. And you see that if you control for contrast, it's still the case that these relevant object contours elicit more activity than those image patches with the same amount of contrast, but that are not part of an object contour. Okay, this is the first indication that this also works. We saw it in texture segregation, that it also works for natural images. Now, I was quite surprised when I looked at the time course. Now it's contrast matched, and you see that these object boundaries already at an early point in time elicit more activity than the not object boundaries. Okay, now we have matched for contrast, but there are, of course, all kinds of other low-level feature differences that are probably going to be different between an object contour and another non-object contour. And it's difficult to completely match for those in natural images. So we do have some indications that this really, maybe V1 cells and cells higher up in the hierarchy are really tuned to those features that define object contours, giving them more activity. Now, there's one thing that we can still do if you really want to see whether this is also a contextual effect, and that's what we did. We took an object contour and we basically transplanted it into the backgrounds. Okay, so now it still seems to be part of the backgrounds. And we can place a receptive field on the original object contour and compare activity to the transplanted object contour that now seems to be part of the background. And if you then look at neural activity, you see that the object contour still elicits more activity than the background contour. But now it happens later. And so now it's really a recurrent effect. Okay, so there's an early effect, probably related to the tuning, and a later contextual effect, allowing the neurons in visual cortex to detect object contours. Okay, so now the last question I would like to address is about region filling. Okay, remember that we had this region filling in texture segregation. Does that also happen for natural images? Now we can still use the Berkeley data set. We compare contrast match features in objects and backgrounds. And basically, all we need to do is do then the normalization for contrast or binning for contrast, and you see the same effect still occurs. Okay, so also image elements that are on the interior of relevant objects elicit more activity than image elements that are contrast matched and are part of the background. And this happens more at a delayed phase of the response, quite compatible with a recurrent influence going to higher areas and then coming back to primary visual cortex, as we saw in texture segregation, actually. Okay, So we see that figural regions in natural images elicit more activity than background regions, even if we match the contrast. Okay, that's good news, because if something does not work in natural images, it's probably not very relevant for perception. It does work. So now I would like to state my overall conclusions. I started talking about figure ground perception. We see that figural regions are labeled with enhanced neural activity. We might call it object-based attention. Boundary detection happens largely in a feedforward matter. Region filling requires feedback. This region filling also depends on attention. And important result for us, if we abolish the late V1 response, we also abolish figure ground perception, linking V1 causally to this perceptual organization process. Then we looked at incremental grouping. We saw that enhanced neural activity spreads according to the Gestalt grouping rules. That thereby to be grouped image elements are labeled with enhanced activity. Incremental grouping is transitive, so sometimes it's very slow. And there's only one label. It's an enhanced neural response, object-based attention. We saw that it generalizes to natural images. We saw that there's incremental time-consuming grouping for natural images. We see that a boundary detection process that seems to be largely feedforward, and a region filling process that may rely more on recurrent connections. And I go back to the very beginning of my talk, where I argued that base grouping is tuning. I did not show a lot of results about base grouping. And then we see there are probably not infinitely many base grouping coded in the visual system, because you have this combinatorial problem, right? There are just too many possible groups that you cannot possibly encode by the dedicated neurons. So you also need this flexible incremental grouping process that quickly finds the right groupings, even for images that you never saw before. And this is my last slide. So I hope you can read what is written here, same or not. And you can probably quickly see that because you have base groupings for the different letters, maybe even for words. Okay, so this goes very fast. But now I ask you, what are the letters that have two dots of the same color? And now you'll find yourself scrutinizing one letter at any one time. Okay, because you don't have base groupings for conjunctions between colored dots and letters in your brain. This is something that you have to build on the fly. This requires incremental grouping. Now, last note. So I've basically been talking about the binding problem all the time. And we see that this is solved by object-based attention, by labeling image elements with enhanced neural activity. These are the people who did the work. I mentioned the people who did the experiments while I was describing them. So I'm not going to go through all of that again, but there are three people I would like to mention specifically. These are Victor Lamme, with whom I collaborated from the start on texture segregation. And he was also one of the inspirators for this work. And Henk Spekrijse, my postdoc supervisor, who was also the editor of Vision Research, passed away in 2003. And finally, Matthew Self, who was involved in basically all the work I've been presenting today. So also big credit to Matthew Self. And these are the funders. So this might be a good moment to start to go into questions. Thank you very much for your attention.
Thank you for a very interesting and engaging presentation. And now we can start with the questions. It's interesting that we have, contrary to our suggestion, that we have very lively activity on Q&A. So maybe we can read the question, especially a few questions that are received the most likes. We see the question from Jeremy Wolf. So he said, in incremental grouping, what happens to the last zebra when you move attention to group the next zebra? Does the grouping of the first zebra persist, or does it fall apart into its pieces?
So this is a very good question, and I think Jeremy actually has a paper on it. It's called post-attentive vision, so he knows the answer. And the answer is that it largely falls back, it falls into fragments. And so if you do a grouping for the same image many times, you may become a little bit faster, but you don't have a lot of benefits. So I actually did a study myself, not very systematically, but I was a subject myself in a curve tracing task for many, many times. And it turns out that I was a bit faster than all the other people. This is anecdotal, but I think if you would train yourself for specific configurations, you might actually get a bit faster. But I think in Jeremy's study, he did not do a training session across weeks. That was just a comparison of accuracy within the same day.
Okay. Another interesting question by Ruth Rosenholz. What happens to the response in higher visual areas like V4 when you silence V1 after the initial feedforward pass?
So we have not tested that, but it's very likely that it is also affected. So there will be definitely the areas that are just after V1, they are probably also going to be silenced. They don't see the input anymore. But what is interesting, because implicitly, of course, you can argue, well, maybe it's not a V1 specific effect. It's just an effect. You silence the entire visual cortex. And therefore, it's interesting to realize that in the contrast detection task, all the feedforward processing in the visual cortex was apparently sufficient. So there it didn't matter. So I think the most interesting result is if you think about that the contrast detection task still worked and the figure ground segregation task did not. So it's not a very trivial result. I think it is really meaningful.
Okay, yet another question by Ruth. Shimon Ullman thought that something like the Groudcon model might work because it might be a crude model of visual crowding. Do you have comments on this?
Yeah, I think that's maybe not the right way to think about it. So crowding has this very special eccentricity relationship. And this Groudcon model, actually Shimon Ullman and Pierre Jolicoeur, they have papers on this, right? So the Groudcon model that I presented, it's not identical to, they also had a zoom lens, an attentional zoom lens. So basically what we find is that the incremental group grows like, I might call it the growing sausage. I'm not sure what is the best way to describe this. And they had a zoom lens that travels along the curve, but they also had the idea that it scales. So if curves are far apart, it's a large zoom lens, and then it narrows it back if curves are nearby. So basically, we're taking over some of these ideas, or they were inspired by the work of Shimon Ullman together with Pierre Jolicoeur and some more of their collaborators.
Okay, the next question is by Benham Karami. From physiological point of view, how it's possible to switch between lateral inhibition in a feedforward mode and lateral excitation in a feedback mode, only in the order of milliseconds?
Yeah, the point is that you don't need to switch. So you can have both processes, and the feedback pathway is basically overruling the lateral inhibition in the feedforward pathway. So that's how we solved it in the model. Now, if you make a model, you have to make many assumptions. So I cannot claim that this is how it works in the brain. I can just tell you that we were able to make at least one model that produces this, and there's no need to switch on or switch off connections.
Okay. Another question by Jordan Dekin. Are the paths traveled by Groudcon retained somehow? For example, if a curve is being explored but then abruptly ends, for example, if the curve is the incorrect curve, is there any residual information that is used to avoid this curve in the future? Or does the process just start all over again with the new candidate curve?
That's a very cool question. So actually you can do all kinds of intelligence tests with curves, right? And I think the answer to this specific question would be is that if you start tracing a curve to find out that it's not the right one, you might use inhibition of return or some kind of suppression process that gives this curve a very low priority, so that while you're exploring the rest of the possibilities, you're initially not very likely to explore this curve again. But this is just speculation on my side, right? I don't have any data to show. So it's a really interesting question, and you could explore this. You could explore this also in in human imaging or just psychophysics. So thanks for the question.
Okay, the next question is by Nicolas Roth. Amazing work. Many thanks for this great overview. Did you check if the center point of the Groudcon corresponds to fixation locations of your participants?
So we did very different variants of curve tracing. So in many of the situations, it's easy to have one of the markers coinciding with the fixation point. So that's just for ease of experimental design. But we also had versions, for instance, in monkeys, where we had the fixation point that was either red or green. And if the fixation point was red, the monkey had to look for another marker that was red, because there was also one that was green, but in that case, he would have to look for the red marker. That was the start of a target curve. Okay, and what you then see is you still get all the same processes at work, but there's a bit of a delay, because the response modulation on the target curve is can only start upon completion of the visual search. Right? So you first have to do a visual search before you can start tracing. We also have experiments in which you first would have to trace before you can start doing your visual search, because at the end of the first trace, you find a color that you then know what you need to search for. So this is much related to the idea of Shimon Ullman, who also talked about visual routines. And I was very engaged by his hypothesis that by having what he called elemental operations, by having a number of elemental operations like tracing, maybe putting something in memory, doing a visual search, and you can make this list a bit longer, that gives you the flexibility to execute all kinds of visual routines.
Okay, next, Andrew Glenister asks, people can also see a white horse among many zebras in the center of a featureless figure. There should be a very little activity in V1 neurons. Do you also expect a process of region filling in to occur in this case, for a featureless figure on a textured background? What would be the point of such filling in?
That's a very good question. Intriguing as well. So there is data on this. So there is a paper by, I think, Klaus Weerhaan, and also Wu Li has papers on it. So if you present, well, there's no such thing as featureless, right? It looks white. So there are neurons in V1 that would code for white. There are not so many of them, but you'll find them if you look for them. Okay, so you can generalize all these things I've been talking about to neurons that code for white. And if you look at figure ground modulation, you actually find it for homogeneous image regions. It's not very strong, but you'll find it. And my suspicion is that if there's a white square, a large white square, you probably find a little bit of this in V1 and V2 and V4 and so on and so forth. But maybe it's not very important, right? Because the edges of the square will allow you to do most of the tasks. But there's definitely something going on there that does not is not incompatible with anything I've talked about so far.
Okay, Balaji, we ask, how will the attention to the figure be affected in a figure ground stimulus in case different grades of blur are introduced, or in a case of different amount of refractive error? Also, will the behavior response correspond with the cortical response in the same case?
I'm not sure whether I completely understood the question. So one was about adding blue.
Blur, yeah.
The color blue, you mean?
No.
Blur.
Like
yeah,
noise.
So your perceptual grouping process is just as good as the input, right? So if you blur away so many features that you cannot distinguish figure and ground anymore, then it just stops. And there is a nice paper also by Victor Lamme, where he made it difficult to see the figures. So at some point, you still see that there are image elements, but the grouping process is disturbed. For instance, if you make the line elements very short, so that it's difficult to judge the local orientation. And then you find some region where the monkeys have a hard time, or your subjects will have a hard time. And he basically found that on those trials where the monkeys could see the figure, very same figure, that you get much more figure ground modulation than on those trials where he did not see the figure. So at some point, of course, you're not able to segregate figure and ground anymore, and then the whole thing collapses. But until that moment, everything seems to be working as you would expect.
Okay, the next question asks Sushrut Torat. When the higher visual areas were silenced in mice, it seems the response to big ground was higher than when there was no silenced. So instead of enhancing the figure response, are the higher level areas suppressing the ground response? A comparison could be made between figure boundary ground to resolve this. Is this activity, is this activity higher for boundary than figure?
So the first part, so I'm not sure how it looked in my slide, but we did a very careful analysis of what happens to the overall responsivity if you silence the higher areas. And the typical finding is that activity goes down a little bit across the board, so it doesn't go up. So this is consistent with an excitatory feedback effect, not so much an inhibitory feedback effect, but there are different effects at different neurons. So there are also papers showing that some of the surround suppression goes away if you silence the higher areas, also in mice, also in monkeys. And we reanalyzed our data, and we do also find these effects. So if you select only cells with very strong surround suppression, then we see if you knock out feedback that it becomes less. So these cells are actually on average excited. But if you take all the cells in V1 into your analysis, then the overall feedback effect is excitatory. So if you inhibit the higher areas, you get a bit of a reduction in activity. That's all in the, all these results are also described in the paper that we published in Science Advances earlier this year about this. There was also something about boundaries, but I forgot, but maybe I already answered the question.
Yes. Maybe to next question, Guido Marco Cicchini asks, great work. I'm afraid I have not gathered perfectly what happens when the object of interest is partly occluded. How does the Groudcon proceed? In the same receptive field by receptive field manner, or does it takes a shortcut using the concept of object?
Yeah. So the answer is already in the question. I agree. So that would be my response. So I presented a version of the Groudcon model where there are multiple areas. And I think that gives the system a great flexibility in applying all kinds of grouping criteria. So some grouping criteria is just because you know what the object should look like, right? So if you see one part of a bicycle that is included in the middle, you see a part of the front wheel, and here's the back wheel, you can probably easily do it by having the activity propagate from early visual areas to cells that code for the shape of bicycles, and then also enhance the rest of the bicycle. But you can also do the same trick for motion by going through motion areas, for color by going through color areas, and so on and so forth.
Okay. Chong Yu He asks, could the rate of region filling be used as an objective measurement of attention?
So region filling, you need to measure region filling, right? So we do that in macaque monkeys, so it's not so easy. So you have your electrode there. You could try it with fMRI, but then you're probably not going to be able to see the time course of it. You could try to do it with EEG. It might work. It's not super easy. And there might be different forms of attention too. So if you really need to know what belongs to what, so for instance, I have this cup, right? I want to grasp it. Now I need to really know what belongs to the cup and what not. So then you really need to do this region filling process very precisely. But sometimes I just need to know what is there. It's the cup or something else. And then I may not need to do it. So I might still direct my attention to the cup, but I don't need to know all the work that is necessary for a very fine-grained segmentation. And in those cases, it might be different.
Okay, next question. Jürgen Kornmeier asks, how is reaction time affected by the size of the Groudcon?
So the Groudcon models basically suggest that every Groudcon shift takes a fixed amount of time. And the logic behind it is as follows. If you have large growth cones, say in area V4 or even area TEO, then these cells are in a map, and you may need to only cross a single synapse to go from here to here. They have large receptive fields, and a single synapse can bridge a lot of space. In V1, where you have tiny receptive fields, you get much more precision, but the time constraints are probably the same as in TEO, but now a single synapse brings you only maybe 1 degree or half a degree, right? So the reaction time does not depend on the size of the growth cone. According to the model, there's just a fixed time delay for every growth cone operation. And if you do this in low-level areas, you just make a little progress. If you do this in high areas, you make a lot of progress. I hope that answers the question.
Okay. The next question, Kagayat Sawyer asks, thank you for the excellent talk. Is there any evidence of region growing or tracing being influenced by memory or more abstract criteria rather than image pixels?
Yes, so of course there are many different forms of memory. So if we talk about long-term memory, then we go in the direction of the first question by Jeremy Wolf, where you can see training effects that might be reflected by altered patterns of synaptic activity. So I could well imagine that if there's a particular curve that has a particular shape, and you're always tracing that you might build feature detectors for that particular shape, and that gives you a number of shortcuts, so you can go really fast across that part of the image. So that would be an answer related to short or to long-term memory. If you think about short-term memory or working memory, it's a good question. Not sure whether this has been tested. So imagine that you have to keep in mind something, a particular shape for another task, and then you have to trace something and it has the same shape embedded into it. It might help, but I don't think people have tested this idea.
Okay, next question asked by Joanna Derbsch. Your presentation argues that incremental grouping is transitive and can be slow. Symmetry perception is usually fast, and EEG studies show is mostly processed in extrastriate areas, V4 and lateral occipital complex, whilst V1 is not active. Could symmetry be considered as an exemption to this incremental grouping being slow that you mentioned?
Yeah, it's an interesting observation. So we also looked at symmetry in our study, and it had very weak effect on V1. So that would be in accordance with the question. And we also actually saw weak effects in area V4, hardly any. Now we don't know how sensitive the monkeys were to this symmetry. So, but in terms of how you detect this, I can well imagine that there are templates in higher areas so maybe feature detectors that are sensitive to symmetrical regions and not so sensitive to asymmetrical regions. So that symmetry might be detected, say, as the letter A or another shape, the shape of a horse, right? And if this is then detected in a higher level, then these neurons that are sensitive for what is now perceived as object could feed back to all those neurons that respond to image elements that belong to the symmetric object. So in that case, indeed, symmetry detection might not be all that transitive. This might be, and this is also, I was talking about Gestalt grouping cues that determine what is perceived as figuring grounds. Not so sure whether those are transitive. And I talked about Gestalt grouping cues that determine what groups with what. And I think there, the term transitivity applies better.
Okay. Next question, Mario Passler asks, does the Groudcon model suggest that there is no need for amplification in V1 for visual field regions that can be grouped by higher level areas alone? For example, if there is just one object in higher level receptive field. Or is there rather that V1 amplification progressed faster in these cases?
Yeah, so the way I described it in the model is that V1 always gets a transcript, always gets the feedback. And if a curve is separated by a large distance from another curve, V1 just looks smart. But you're actually looking at V4 processing that is sent back to V1. Now, the advantage of this situation is that you never know when you enter into a particular hard region of the image where curves are nearby. And that's very nice that V1 already knows it. So now the higher areas are knocked out because they can't do it anymore. And V1 got the feedback from the higher areas, so can take over for a while. And once you're past this difficult region, then the higher areas can take over again. That's the way I think about it. But I must admit that I don't have any strong evidence in favor of this idea.
Okay, thank you. I think we covered all questions, as I can tell. So we can now conclude the session. Thank you, Pieter, for a great presentation, and thanks all participants for listening this. So now we can go to a short break before the next session that will be here on perception and action. So thank you again to all you.
Bye-bye.
Goodbye.
Thank you, Peter. So Drajan, the next session starts at 3 o'clock CEST.
3 o'clock.
Correct?
3 o'clock.
Yeah.
Yes. Sorry.
We're beautifully on time for a half an hour lunch or breakfast or dinner break, whatever we have. So see you all later.
Okay. See you at 3. Bye.